Dear learners, welcome to APCC LMS portal. I am Dr.N.Ankamma, Assistant Professor in Zoology from Government College for Women, Guntur. In today's class, let us discuss about system of inbreeding and crossbreeding. Before going to start the lesson, let us see the learning objectives of this lesson. At the end of this lesson, you will be able to understand the importance of animal breeding, role of breeding techniques to improve the dairy production, importance of inbreeding, role of crossbreeding, difference between inbreeding and crossbreeding. All these things you will be able to learn at the end of this lesson. Now let us see what is inbreeding. Inbreeding is a mating system in which progeny produced by the parents are more closely related than the average of the population. So inbreeding is a result of mating of the individuals which are related to one another by having one or more ancestors. So inbreeding is the mating of closely related individuals. It refers to the mating of more closely related individuals within the same breed of individuals in a lineage. We can explain inbreeding by taking an example of cows and bulls. Let us take superior cows, Bos indicus, and superior bulls, that is Bubulis bubulis, of the same breed are identified and they are mated. Here the progeny obtained from such mating are evaluated and superior males and females are identified for further mating. If we see the classification of inbreeding, inbreeding is classified into two types. One is close breeding and another one is line breeding. If we see close breeding, the name itself is self-explanatory. That is, in this type of inbreeding, mating is made between very closely related individuals, such as own brothers are crossed with sisters or offsprings are crossed with parents. That means male parent, which is called as sire, is crossed with female offspring, or female parent, that is dam, is crossed with male offspring. So here closing breeding, mating is between very close relatives, that is brothers and sisters or parents and offsprings. Second one is line breeding. If we take the line breeding into consideration, here mating of distantly related animals that had a common ancestor, for example cousins, half brothers, sisters, granddaughters and grandsires. What are the advantages of this inbreeding? Why this inbreeding is practiced in dairy technology? So inbreeding, it increases homozygosity. Here homozygosity means both the genes of the allele. It may be recessive genes or it may be dominant genes. If they are same, it is considered as homozygosity. This inbreeding increases the homozygosity in the offsprings. So as the inbreeding proceeds without selection, a large population becomes a series of subpopulation, and inbreeding brings to light the hidden recessive genes, because recessive genes can be expressed only in homozygous condition, and this inbreeding increases the homozygosity. This inbreeding brings out the hidden recessive genes in the animal population. Genetic makeup of unknown or non-descriptive herd can be determined by this inbreeding. As a consequence of increased homozygosis, the characters are fixed in a inbreed population due to gene fixation, and inbreeding increases the prepotency in the animals. Let us see the disadvantages of inbreeding. Inbreeding may express harmful recessive alleles and the phenotypes concerned, because it increases the homozygosity. When the recessive genes are harmful, these recessive genes may be expressed in the generations. Continuous inbreeding, especially close breeding, which is made between very closely related individuals like brothers and sisters and parents and offsprings, it usually reduces the fertility and even the productivity, which ultimately affects the yield in the animals. As per Darwin, the consequences of close interbreeding carried on for longer time are generally believed in loss of size, constitutional vigor in the animal, fertility of the animal, and sometimes accompanied by tendency to malformations, which may be a major loss for the animal breeders. Inbreeding result in the decrease of fitness state and also result in reduction of production rates. Since there is an increase in homozygosity, may be a chance for the appearance of lethal genes. These are the major disadvantages of inbreeding. And sometimes this inbreeding also leads to inbreeding depression. What is meant by inbreeding depression? If we see, it is the phenomena of a reduction of fitness trait in the animals. The decline in average phenotypic performance of an individual is called inbreeding depression. Increased level of homozygosis is the underlying factor which causes inbreeding depression. So major cause for this inbreeding depression is increased level of homozygous due to continuous inbreeding. An inbreed individual is more likely to be homozygous for any gene. So the animal is likely to express undesirable genes and hence the undesirable traits. So inbreeding depression is essentially the opposite effect of heterosis or hybrid vigor, which we will see in case of crossbreeding. These are some of the points related to inbreeding depression. So inbreeding depression ultimately leads to reduction in the fitness traits of the inbreed individuals. What are the effects of this inbreeding on reproductive performance of the individual? If you see, it slows the testicular development, that is the male reproductive structures from which male reproductive gametes are formed. There will be a slowing down of the testicular development due to continuous inbreeding. It delays the puberty, the age at which the animal reaches reproductive potential. Next, decreases in the number of ova shed by the females. It increases embryonic death rates. All these are the effects on reproductive performance of the animal due to inbreeding. And inbreed animals are more susceptible to critical environmental conditions like drought or lack of certain sufficient food materials, fodder. All these conditions will be a critical environmental conditions than the non-inbreed animals. And inbreeding causes the appearance of genetic defects in the offsprings. In general, inbreeding results in overall lowering in the performance of the animal, vigor of the animal, reproductive efficiency of the animal, and ultimately survivability of the animal. For instance, the spread of spider lamb disease in block-faced sheep is believed to be the consequence of inbreeding. This is the spider lamb disease in black-faced sheep, in which shape of the here the face is black in color. That's why it is called as black-faced sheep. In this black-faced sheep, due to continuous inbreeding, it causes a disease called spider lamb disease. This is the main drawback of one of the example for defects in inbreeding. And what are the uses of inbreeding? Inbreeding is used to develop seed stock. Inbreeding is used to test a sire for an undesirable recessive gene. Inbreeding promotes an increase in prepotency in the animals. Inbreeding is used to uncover the genes that produce abnormalities or death so that it can be removed from the population. Inbreeding is used to produce distinct families within a breed, and inbreeding with selection had helped in past to develop several breeds of livestock like longhorn cattle, Leicester sheep, and Shire horses. So this is the longhorn cattle which is developed through continuous inbreeding. This is Shire horse and this is Leicester sheep. So all these are the products of inbreeding. Now let us see the second type of breeding which is just opposite to inbreeding, that is crossbreeding. Here, if we see the definition of crossbreeding, the mating of animals from different established breeds is called as crossbreeding. Here, the progeny produced is called as crossbreed. For general usage, it might be assumed that a crossbreed was the result of planned mating. What are the aims of this crossbreeding? Why the crossbreeding is uh, allowed in dairy animals? The aim of this system of breeding is to use the best sires available regardless of breeds and to gain hybrid vigor in the offspring and possibly more economical milk production. So the main purpose is to use the best sires and to get hybrid vigor. Hybrid vigor means if the performance of the offspring is better when compared with the average performance of the parental generation, this can be called as hybrid vigor or heterosis. It is the most useful phenomena which we are getting through crossbreeding and increase productivity over state breeds because of resulting hybrid vigor. It produces commercial animals with desired combinations of traits not available in any one breed. So, ultimate purpose of animal breeders is to produce commercially more beneficial animals with the desired characters which they want, which is not available in any one breed. We can by crossbreeding, we can get all the desirable characters in one breed. What types of crossbreeding? The crossing of different breeds can be practiced in different ways. It depends upon the number of breeds used and the manner of crossing. So the major forms of crossbreeding are one is regular crossing and another one is composite crossing. Now let us see one by one. Here regular crossing means a process in which mating of the same cross on regular basis to take the advantage of heterosis as well as complementarity. This is also called as systematic crossing. So here the crossing exploits non-additive gene effects through heterosis and additive gene effects through complementarity when two or more characters complement each other. There are two different methods of regular crossing again. One is specific crossing, second one is rotational crossing. Now let us see one by one. This specific crossing is again of different types depending upon the number of breeds used. One is two breed crosses, second one is three breed crosses, third one is four breed crosses, or sometimes even five breed crosses are also there. This can be further grouped into the following forms. In case of two breed crosses, two pure breeds are crossed together. This includes two pure breed crosses, interseminating, back crossing, and criss crossing. In case of two pure breed crosses, crossed progeny are not used in breeding programs. Whereas in case of interseminating, this is the crossing of crossbred progeny having the same level of inheritance of two breeds like crossing of F1 with F1. By crossing, let us take an example of breed A is crossed with breed B. We are getting progeny F1. In the progeny F1, one male and another one is female. If we again cross the F1 individuals, F1 male with F1 female, it is called as interseminating. And next one is back crossing. Back crossing means it is the mating of crossbred animals to purebred animals of either breeds used to produce progeny of first generation. That is, F1 is crossed with the parental generation P1 or F1 is crossed with parental generation P2. Where here P1 and P2 are the pure breeds used to produce F1. So backcrossing is a crossing in which F1 individuals are crossed with either of the parents. It may be a F1 female is crossed with parental generation of male, or F1 male is crossed with parental generation female. This is called as back crossing. If we take the bull of breed A is crossed with cow of breed B, which are pure for their breeds. When there is a cross between these two, we are getting the F1 progeny, that is called AB. Here it is getting 50% characters from A and the remaining 50% characters from breed B. If we take three breed cross into consideration, in this system of crossbreeding, three breeds are used. So, the first cross animal is mated to animal of third breed like AB female and C male. When breed 1 and breed 2, let us consider it as A and breed 2 as B. When there is a cross between A and B, it leads to the progeny AB. This is F1 generation. When this F1 is again crossed with breed 3, let us imagine it as C, we are getting F2 generation. So here why it is called as 3 breed cross means here breed 1 and breed 2 are crossed to get F1 generation. This F1 is again crossed with breed 3 to get F2 generation. So we are using three breeds, breed 1, breed 2, breed 3 to get F2. That's why it is called as 3 breed cross. Next one is 4 breed cross. It is also called as double 2 breed crosses. This involves the crossing of crossbred females produced by crossing two breeds A and B with the crossbred males produced from crossing another two breeds, that is C and D. Thus the mating is between AB and CD. Let us imagine this is A and this is B. When there is a cross between these two breeds, we are getting F1 generation, which is AB. And this is one double cross. In next case, we are taking this is C, which is pure breed. This is D, which is also pure breed. When we cross these two, we are getting the F1 from these two breeds. That is, this is also F1 from C and D. Again, this is the product of A and B. This is the product of C and D. Ultimately, again, we are crossing this AB with CD. We are getting the F2 generation. These two are F1, and when there is a cross between these F1s from different breeds, this is from A and B, this is from C and D. Ultimately, we are getting F2. That's why it is called as 4 breed cross and also called as double 2 breed cross. So ultimately, AB and CD are crossed together to get F2 generation. And another crossing is rotational crossing. The males of 2 or 3 breeds are used in regular sequence in successive generations on crossbred females of the previous generations. This involves 2 or 3 breeds. The major disadvantage of this rotational crossing is that it does not allow any exploitation of complementarity, but it shows only heterosis. So the disadvantage of rotational crossing is it does not allow the exploitation of complementarity, but it shows only the heterosis or hybrid vigor, where offsprings has better characters than the average characters of the parents. If you see the rotational crossbreeding in cattle, A and B. Again, the F1 is crossed with A, F2 is again crossed with B. See, let us imagine this is breed A and this is breed B. Due to the crossing of breed A and breed B, we are getting the progeny. In the progeny, 50% A characters and 50% B characters. Again, the progeny obtained through A and B crossing is crossed with one of the parental generations, that is B. And the progeny obtained through F1 and B is again crossed with parental generation A, and the progeny obtained through this cross is again crossed with B and A and B. So ultimately it is simply a rotational crossing of progeny with the parents. So starting at 50%, 50%, the rotation stabilizes at 65 by 35% or 35 by 65%, giving 65% from the last sire line used. This is called rotational crossing. So final day, inbreeding, it only hurts the children's children. So continuous inbreeding leads to accumulation of recessive genes. Sometimes these genes may be a lethal genes. So inbreeding, if it is practiced continuously, it may cause lethality also. Thank you.